all. This is Dr. Mubin Sayed from drbeen.com. Welcome to one more show. So today we have a fascinating study to share. It is from Harvard University and Cairo, Egypt. Beautiful study. I loved it. When I was reading it, it was just tickling my fancy. So let's look at it. So starting from my latest painting, this painting's title is Your Memories. It is uh, painted in Rebel 5, which is a digital painting software. This is drbeen.com. In the description of this video, there is a link where you can get, with just a limited time, a $67 access to everything in drbeen.com. And this is not a recurring payment. Then here is the study that we're going to talk about. Cold exposure impairs extracellular vesicle, EVs, swarm-mediated nasal activity, antiviral immunity. And if you are here that EVs are electrical vehicles, you are welcome as well. We do have EVs in our nose and actually all over our body, especially in all wet surfaces. So we're going to talk about the EVs today. So you're welcome. This is a study. It is behind a paid wall. So I have the study's PDF. I paid for it and got it, but I cannot go over the whole PDF with you because of the copyrights. Then there are some more articles as well that may help look at it. So now let's start. I think you will like the diagrams that I did for this one too. So here, the basic idea is the following. Our epitheliums are the surface cells, especially the mucosal epithelium, that is the wet surfaces and the cell, and then especially the nose. Our nasal epithelium is protected by swarms of EVs, extracellular vesicles, and cold reduces the functionality of these EVs and the number of these EVs. That allows a virus to infect us more easily. So that is a basic summary of it. Let's see more. I made this little diagram. Here there are some chilled out, frozen out EVs. And here is the swarm, hornet's nest, if you will. And we are going to connect these two images in our discussion. These are our gifts for humanity. They're continuing. This is a study that I just discussed. And then what I want to do is, first of all, let me discuss this. We know that there are many mechanisms due to which we become a victim to cold. For example, vitamin D matters, especially in the cold times. We have less sunlight in many geographies. Then people have bundled themselves up in clothings that also reduce the sunlight to the body. So vitamin D is an important aspect. Viruses replicate better in cold environment. For example, coronaviruses, they actually like relatively colder areas, so that is throat and nose. Although SARS-CoV-2 was a virus that decided that, hey, I would love to replicate or increase in number even at a warmer temperature, and that is why it was able to go down in the lungs and still replicate, or even in other tissues as well. But viruses, bacteria, they do like colder environment as well. Then folks with comorbidities, immunocompromised, transplant patients, patients on drugs, they can also fall victim to more infections. Socioeconomic status, hygiene, moisture, environment, that also matters. Many other facts are out there as well. Many other factors are there. Then, of course, this is a new one. You would love it. I'm sure that you would love this one. Then, of course, we know innate immunity is important. IgA that is present, these are the antibodies present in the wet surfaces of our body. Complement system, mucociliary interface. So at least in the respiratory system and in our nose, there are cells which are wet, and then they have tiny hair like projections from them that beat around and they move things, and that is mucociliary interface or escalator. Then there is mucus itself, the wet surface itself washes out the pathogens and more. So there are many factors. However, let's look at this new factor we found. So for this discussion, keep in mind a hornet's nest. Imagine you see a hornet's nest and there are some hornets that are, you know, flying around it. But if you kick the nest, then there are thousands or hundreds of hornets that would come out and attack the offender. That is exactly what happens when a virus dares to kick the nasal mucosa. So here, imagine this blue cell is our anterior nasal epithelial cell. Anterior nasal means the interior part of the nose, the front of the nose, and the epithelial cell is the surface cell. And here it has this TLR3. So the cells, immune cells and normal cells, non-immune cells, normal civilian cells, they can have receptors on them called toll-like receptors that can match or recognize pathogen patterns. And once they recognize a pathogen, they can then react to it. Our nasal mucosa, anterior nose, when that is exposed to a virus or bacteria, the toll-like receptor 3 receptor on it becomes engaged with the virus or bacteria and becomes activated. The result of that activation is that many tiny pieces are pinched off of the cell. These are the extracellular vesicles that are formed, EVs that are formed. So we have done this discussion a few days ago with the mother's breast milk having messenger RNA in it in context of the COVID vaccines. So that was also extracellular vesicle. Extracellular vesicle is nothing but a small envelope of the cellular material that has pinched off or separated from the cell. Now, interestingly, so these EVs here, these little orangish pieces, interestingly, our nose does something very interesting. It gives these EVs more receptors to bind with the viruses. Nowadays, we've been talking about COVID a lot or SARS-CoV-2 a lot. So let me give you an example of SARS-CoV-2. Imagine that we know that SARS-CoV-2 binds with ACE2 receptor. So imagine when our nasal epithelium makes an EV, it will give that EV a lot of ACE2 on it. That EV now will become a decoy, or decoy, whichever way it is pronounced. It will become a decoy to bind the SARS-CoV-2. That is the interesting thing. So these EVs, they don't have a nucleus in them. They don't have the cell's machinery in them. They have a high density of receptors that can be used by viruses. And inside the EV, there is microRNA. MicroRNA is known to be antiviral. It's such a beautiful poison our cells make for viruses. So we make these EVs all the time. But if the TLR3 is engaged with the virus, then these cells start making a lots of EVs in billions in the anterior nose. And these EVs will then start engaging. Now, before we go there, please realize that the receptors on the EVs are about 20 times more dense compared to the regular cell. What is the cell trying to do? The cell is trying to say that, hey man, virus, you are here to attack us. Why don't you attack this little thing? And the virus, if attacks that little thing, the EV, the virus is going to end up in a dead end. Why? Because that EV is going to be empty. It's not going to have a nucleus. It's not going to have a machinery that virus can use to make more viruses. Plus, inside the EV is the poison microRNA that can kill the virus. So really, the virus is in trouble. And that is how we protect ourselves during normal, regular temperatures. When the temperature drop, then something happens, which I'll discuss in a second. So first thing, the receptor density on the EVs, receptor for viruses, is 20 times more. Then, second thing, that when a virus engages, so imagine these little dark orange things are viruses. When they engage and they cause TLR3, then these EVs, as they emerge, the whole swarm of EVs, billions of EVs are released from the nose into the mucosa, into the wet surface of the nose. And these EVs start binding with the cell. Inside the EV is the microRNA. MicroRNA is a tiny RNA, as the name says, microRNA. This little microRNA travels in the EV to another cell and brings a message to the cell. So this is one cell sending a message to another cell. And what is that message? It can tell the other cell that, hey, you need to bring your defenses up and you need to start having gene expression or making proteins to protect against the viruses. At the same time, this microRNA can actually cause the target RNA of the virus, for example, to break down. And do you know how it does it? It's such a fantastic thing. It causes hydrolysis of messenger RNA of the viruses or the cells. So what happens is hydrolysis means it puts water on it. So imagine you are cooking something delicious and tasty, and someone comes along and with a bucket of water and puts all that water in that delicious, tasty food. All of a sudden, that food is now not delicious anymore. Same is the case that when the viruses and their mRNAs are trying to work, and the EV comes along and brings microRNA, which causes hydrolysis of this mRNA, the mRNA just breaks away. Similarly, an mRNA, messenger RNA of the virus or another cell that needs to now become translated into a protein, which may become a virus protein, microRNA can stop the translation of another mRNA. So it can stop production of new cells. It can stop production of new viruses. So it is a virucidal drug. It is a drug that we make that kills the viruses. EVs carry that little drug. And if a virus becomes attached to the EV and enters the EV, then the virus has entered a dead end. It has entered a jail. Inside the EV, the only thing it would find is some microRNA that is going to kill the virus. The virus is not going to find a nucleus to work with. It is not going to find a lot of ribosomes to work with. It doesn't have the machinery to replicate. This virus is trapped in a jail and it's going to die. And more viruses would enter EVs compared to the normal cells because EVs have a lot of virus receptor, 20 times more receptors. And similarly, EVs have 10 times more microRNA compared to a normal cell. So, what happens normally? When a nose is triggered, induced by the presence of a virus, or nose cells, epithelial cells, they produce more EVs. Those EVs start mopping up the virus because they have more receptors on them. The viruses get trapped on the EV and also they go into the EV where the microRNA start killing the viruses. This is a normal behavior, beautiful mechanism. And this was known before. So this is not a new thing. The swarms of EVs attacking the viruses was known. But now check it out. What happens in the cold time? In the colder environment, so the researchers did this research. They reduced the temperature, or they exposed some people, healthy people who did not have any infection, who also did not have any allergies, and were healthy when they were being tested. So what they did was they exposed them for 15 minutes to 40 degrees Fahrenheit or 4.4 degrees centigrade, just 15 minutes. And that reduced their temperature inside the nose by 9 degrees Fahrenheit. And guess what happened? These little decoy cells, these little EVs, these fake cells that we had made for the virus to become captured in, the jail cells, half of them reduced. Not only that, 50% reduction in microRNA inside the EVs. So when the newer EVs were created in this cold time, microRNA injected in them was 50% less. So that means that the cells that became cold and were trying to make more EVs in the presence of a virus, these could not make enough microRNAs for them, or could not give them enough microRNAs. The receptor density on the EVs dropped by 70%. So again, a cell that is now operating in cold and is trying to make an EV because a virus is stimulating the cell to say, I am going to attack you, and the cell is trying to make these decoys, the receptor density on these decoys is 70% less. 42% of the EVs got knocked out. Existing EVs, already formed EVs, 42% got knocked out. So half of the immunity, this is almost like innate immunity. It's a different kind of innate immunity. Almost half of the innate immunity dropped. And that would then allow the virus to more freely infect and cause disease as well. So disease is the production of symptoms. Infection is just presence of the virus. So this is a study that is in vitro plus ex vivo. What does that mean? The whole experiment was done in vitro in the petri dishes, but the cells were taken from the healthy individuals, and those cells were then cultured outside. So that is called ex vivo. So again, what happens really inside our bodies would still need to be seen, but this is a beautiful mechanism. And so then what is the takeaway? Of course, there were lots of jokes people were making that, you know what, we should start making nasal socks. And I Googled it, and it turned out there are actually nasal warmers, nose warmers or socks. So anyways, it looks really strange. So somehow to keep the nose warm, that is with a muffler or whatever way you can, then they think that in the future, there will be drugs that would poke the anterior nasal mucosal cells to cause production of EV swarms. And these swarms will then become protective layers. And as the actual viruses come and attack, these layers would then mop them up, engage them, trap them, and kill them. This is a similar behavior that we enjoy during non-cold time. We don't get that much of cold or flu during non-winter times. So we could, during the winter, have drugs that would help do a similar thing. And more on a personal level, I have seen, at least for myself, again, it's not necessary, it is for everyone, plus it's not a prescription. I've seen more on my level, nasal spray has been really helpful. So nose warming, whatever mechanism, a mask, a nose sock, or just not going out in the cold, or having a muffler wrapped around the face area, or nasal sprays, or maybe some medicines in the future. So this is the discussion. I really, really enjoyed this paper. Let me just give you a glimpse of this paper once more. Harvard and Cairo are the researchers from, and here is the EV discussion. So someone said, Nawaj, sure, Lavage as well. M. Gregory says, please no more drugs. Sure. So there are any way you can keep your nose warm, at least in light of this discussion, can help. So there is a Primal Colin says, nose sock is worth of a picture, Dr. Bean. I'll have to buy a nose sock and make a picture. So this is the discussion. Please like, subscribe, and share. There are links in the description. If you would like to support this work, you can buy me a coffee, you can use PayPal, you can become a patron, and I will see you tomorrow. And by the way, today I did a discussion for FLCCC as well, which is a real fascinating discussion that it is about photobiomodulation and inflammation. So hopefully that will be published from their platform in a very near future. So with this, thank you very much. I will see you tomorrow. Bye for now.